Thank you so much for giving me the opportunity to talk today. It's kind of cool to be with a group of people where I probably have more than half of you on my Google Scholar news list, and I read so many of your papers. And I'm excited to talk about some of the transcriptomic data that I've gathered over the last couple of years. So we're kind of doing a little switch away from the functional, looking into transcriptomics, and hopefully at the end bringing it just shortly back to functional. So I've looked at kind of the neuropathological spectrum that underlies medial temporal lobe epilepsy, something that we've looked at, I think everyone here, the tissue that we're using. So just to give a little bit of a reminder of the background, medial temporal lobe epilepsy is the most common form of focal epilepsy, where seizures start within the hippocampus. And devastatingly, 80% of these patients are pharmacoresistant. We all understand how severe these seizures impact the patients, but I think it's a good reminder that we really need to understand more about this disease. The hippocampus undergoes significant changes. There's a loss of neurons in specific subregions, granular cell dispersion, as well as mossy fiber sprouting. But it's really been the loss of neurons in specific subregions of the hippocampus that has been used to further define MTLE into specific subtypes. So the superheroes of the International League Against Epilepsy in 2013 have used the cell loss to define these ILAE grades. So we can see in ILAE 0, it's classically thought of the gliosis-only type, where there's no neuronal cell loss, while ILAE grade 1 shows CA4 and CA1 prominent cell loss, which you can see here by this absence of the staining of the neurons. And then ILAE grade 2 shows CA1 prominent cell loss, and ILAE 3, which is the most rare form, shows CA4 prominent cell loss. So this has been really helpful to group patients and to begin to understand MTLE a little bit better. But I wanted to dive deeper into it and understand distinct gene expression changes at a single cell level with intact tissue to hopefully understand, are these discrete types? Do we see common different gene expression changes between these types? So I've used single cell spatial transcriptomics, and specifically a method called Xenium by 10x Genomics, which is in situ based. So we get our tissue blocks, so I've used FFPE as well as fresh frozen samples, and then sections are placed onto this very special slides, and then we hybridize probes onto the tissue, which is followed by staining for cell bodies as well as nuclei that helps with cell segmentation. And then once all this bench work is done, we load the slides into the Xenium machine, which does everything by itself, so it's super nice. And it goes through iterative rounds of fluorescent labeling of specific probes, imaging, stripping, labeling again, and so on, until each probe that we wanted to measure has been imaged, and we can get the data out of it. And I've used a probe set of, in total, 366 probes that contains marker genes for different cell types, as well as epilepsy genes that I've found in literature, most of your papers actually, probably. And to give you a bit of this doesn't work, idea of what this looks like. Here I show you the output data. So each dot represents a transcript, and I've only shown you 15 of the 366 transcripts here. Of a small section of the tissue, but you can appreciate that we can now identify the specific location of individual transcripts and associate them with segmented cell bodies. So this is here also DAPI staining. So I've done analysis in R using C.E.T.S, for those who want to know, ran normalization as well as harmony integration to reduce batch effects, and dimensionality reduction, clustering, and differential gene expression. And using this, I was able to characterize more than 1.1 million cells from 18 human hippocampus samples, and which you can see that in this kind of cool animation that all of these 18 samples we know exactly the spatial location of the individual cells, but they all integrate well together into this one UMAP. Just to give at the second day a bit of a background, what is a UMAP for the 5 people in here who might not know? Each dot represents one cell, and cells that sit together in space and that cluster together so they have the same color express the same genes, so they have the same molecular profile. And if we look at canonical marker genes, we can then identify which are the underlying cell types. So overall, like broadly speaking, I was able to identify all the big cell types in the human hippocampus: oligodendrocytes, excitatory neurons, inhibitory neurons. I next wanted to look at just general proportions of these cell types, if we have them in all patients, and we broadly do. And one immediate interesting fact, which is a good, was a good sanity check at the beginning, if we look at the proportion of excitatory neurons, we can see this kind of split between samples that have no hippocampal sclerosis and samples with hippocampal sclerosis. I forgot to mention the P is these are postmortem samples that I used. These are from patients that have no history of seizures or epilepsy. I then ran differential gene expression at this broad level to get an idea how differentially expressed are genes overall between cell types. So for each cell type, I did a pairwise comparison between two specific groups. And in the bar graph here, I've plotted just the number of differentially expressed genes, and the lighter colors, like too far to the right, show genes that are upregulated in this, like in the column groups versus the row groups, and those that are darker are downregulated. And one thing that was kind of striking to us is that we see very, very few, nearly no differentially expressed genes in our panel between ILAE 0 and postmortem control. So these are epilepsy samples versus samples without epilepsy. However, we see lots of differentially expressed genes between both ILAE 1 and ILAE 2 compared to our controls. Again, when we compare ILAE 1 and 2, so both hippocampal sclerosis samples, to epilepsy samples without hippocampal sclerosis, we see less differentially expressed genes. And when we compare ILAE 1 versus ILAE 2, so the two hippocampal sclerosis groups, we again see very few differentially expressed genes. So we hypothesized that hippocampal sclerosis is really the main driver of transcriptomic remodeling in these samples. Next, I wanted to take a closer look at excitatory neurons, because I think those are the cells that we've heard about today or yesterday the most. And I kind of like subsetted just the excitatory neurons, did my reclustering again, was able to identify all these wonderful cell types that we all know from the hippocampus. So we have granular cells, CA2, 3, two CA1 cell types, as well as 3 subiculum cell types, all expressing canonical marker genes that we know from literature. And when we plot these in space, we begin to see this really beautiful structure of the hippocampus, our dentogyrus granular cell layer, and then our trisynaptic loop, with in the subiculum these subtypes that we identified really nicely layer from superficial to deep. And next I wanted to see, so we can already see that we have this like cell loss of excitatory neurons in regions that we know they should be lost, because that's the ILAE type. So we have loss in CA1 as well as CA4 in ILAE1 and only CA1 in ILAE2. For those who are missing ILAE3, they are really rare. I only have 2 samples, so I haven't included them yet. But I wanted to give this a little bit more numbers. So I looked at the density of cells for each cell type within the samples, and want to highlight just the difference between the 2 CA1 subclasses that I've identified. So I found a significant decrease in deep CA1 cells versus superficial. In superficial CA1 cells, I did not see this decrease in epilepsy, in samples with hippocampal sclerosis. And just for those who don't know where the deep and the superficial CA1 cells would sit, so deep in the hippocampus is an area that is closer to the alveolus, so to the outside, and the superficial is more to the inside. It's the way that cortex versus what I found is that the specific cell loss is really associated with a CA1 region that is a distal CA1, a proximal CA1, so closer to the CA2. And superficial CA1 cells tend to sit close to the subiculum, as you see these yellow cells here. So we think that cell vulnerability is not just driven by cell type, but it's driven by spatial location, likely because of network connectivity within a specific region of the hippocampus. And a really wonderful student in the lab, the next cell type that he wanted to look at, Adi, was microglia. So he subsetted the microglia to look at microglia states in mesial temporal lobe epilepsy. And he identified multiple microglia states that express specific canonical marker genes, and really highlighted 4 known groups: the homeostatic microglia, disease inflammatory macrophages, border-associated macrophages, and disease-associated microglia. Really a mouthful. Yes, let me have a break. When you live in Canada, you learn to just patience your way through these. So the next thing that Addy did is he plotted these 4 big groups that he identified in space, where you can see really nicely. It's all you can see, you don't have to hear me. See these blue dots that associate really nicely with regions of hippocampal sclerosis. So to kind of like recapitulate this dramatic infiltration of these specific subtypes, he wanted to see if he can also identify broad gene expression changes in the overall microglia population. So he pseudoblocked and then looked at canonical marker genes for DIM, homeostatic, as well as DAM. And he was able to see that there's a significant upregulation in ILE1 with marker genes that are associated with this specific state of microglia. And while he also saw a upregulation of homeostatic microglia markers from ILE1 to ILE2, and no effect in marker genes with DAM-associated microglia. So one thing that we've found in all of our data analysis, so I'm not showing all of it, but we saw this trend of gene expression changes between like non-hippocampal sclerosis versus hippocampal sclerosis, but even within hippocampal sclerosis from ILE2 to ILE1. So the more cells that are lost, somehow we see more regulation. So we wanted to conceptualize this a little bit more, what I did is I averaged the gene expression for all cells. Like basically I made one model cell for each patient to then compare the molecular profile of each patient across all patients. Which is why we now end up with 18 dots, and then I did principal component analysis, which gives us an idea of kind of which genes have the highest impact on variability between samples. So PC1 is the principal component that explains 30% of the variability, which is pretty high. And you can see that samples are kind of shifted along PC1 to somewhat divide between samples with hippocampal sclerosis versus without hippocampal sclerosis. So I took a closer look at the genes that are specifically involved in this loading and in this shift, and I've identified cells that are involved in synaptic transmission and excitability, as well as oligodendrocytes and myelination and microglia homeostasis. All of those are involved in kind of the positive loading, and the negative loading we see a lot of immune signaling, as well as extracellular matrix remodeling, which I think is something that's also really important. Taking a closer look at our top loading, KCNQ3, I was able to show that there's a specific upregulation from ILE2 to ILE1 in dendrite gyrus granular cells. I haven't gone into detail into the analysis of these. There's like subclusters that I've kind of like hidden from you all. I think this would be its own kind of paper, which is really exciting because that's kind of what this kind of data can do. Can give it out and then do even more analysis. And I just want to highlight that Xenon, which is a Vancouver-based company, has recently done a clinical trial too of a KV 7.3, which is KCNQ3. They have a channel opener that works really well in focal epilepsy patients that are pharmacoresistant. So they put this out, and then we found this in our data. It kind of like works really nicely together. So to summarize this, I love this. I've shown you that there's region and cell type specific gene regulation, which we can identify with single cell spatial transcriptomics. I have a really cool video going as well, actually. And that proximal CA1 seems to be more vulnerable than distal CA1, and that there's an infiltration of diseased inflammatory macrophages that seems to be specific to regions with neuronal cell loss. And if we look at kind of like the molecular phenotype of individual patients, we can identify a graded continuum of gene expression. We've also run some really cool domain mapping using precast with another grad student in the lab, as well as interneuron. We've shown like interneuron subtype remodeling across ILAE grades. So there's lots of data still to come, and we're going to put this out for everyone to investigate later on after I've published it. So I have to think about my postdoc, okay? One thing I want to mention at the very end, so this is all transcriptomic data. For all the functional people in here are like, oh my God, this FFPE tissue, where's the living stuff? So in the Samrowski lab in Vancouver, and as well, I've done this, I've done a lot of functional investigations in my PhD as well. We also have slice cultures, and we use calcium imaging to look at different types of activity. Julia's presented her wonderful poster looking at AD and effects of A beta on neuronal excitability. And I just want to highlight that with all this establishing over the last 5 years, we now have a platform that is generously supported by Brain Canada, which is called the Gene Cell Circuit Advanced Neuroscience Platform, so UBC GC2. And it's intended to advance technologies to explore brains at multiple levels, from molecules to behavior. And it's really supposed to be a sharing platform where we can do experiments. Someone comes, like researchers come in, you come in with cool ideas, and then we can do them together there. Mevish is here today. She's the coordinator of the platform if you ever want to have more of a chat about it. I just want to highlight some of my data. Some calcium imaging that I've done where I induced epileptic activity with Hannah. We have to talk about this because I see deep layer kind of like being recruited or starting before superficial layers. And then what I really want to do, is what I've started, is to follow this up with MFISH, where we can look at different types of inhibitory neurons that are involved in this activity at really a single cell resolution. And I mean, the dream is to do this with spatial transcriptomics after, and I'm hoping that we can not only expand the size of transcriptomics, but also the thickness, so we can do this in a full 300 micron slice. That would be really nice. And yeah, with that, I would like to thank my absolutely wonderful lab, my professor, my PI, Mark Sambrowski, and the neurosurgeons that we work with. Cannot highlight more neurosurgeons as well as Mo. It's amazing. Our neuropathologist, John, and the students in the lab that helped me with my research, and our really great and generous supporters and funders. Thank you.
Thank you for a really great talk. Very, very interesting. Do we have time for a few questions?
I really appreciate the xenium data from the human hippocampus in these disease cases. Maybe I'm reaching too much, but I really enjoyed that study I know you were part of on the description of the ovoid cells in the deep layer subiculum in the rodent. Was I hallucinating, or in that last slide, do you see something interesting going on? Because with the spatial data and the
Mark is so excited about that, yes.
You can see that type popping up with the KCNQ3?
Here? Maybe.
And it's a conserved type, obviously, from rodent to primate.
It is, yeah. So I've actually identified those as well in the deep subiculum cells right here. So they said, yeah, so we see them. They have like CHARM2 expression, and yeah, we're investigating those as well. For those who don't know, my supervisor, Mark Sambrowski, has identified a deep-lying subiculum neuron. We call them ovoid neurons. They have really freaky morphology. They're super hyper-excitable. They're sustained activity, which is absolutely bonkers, and they're involved in novel object recognition. And we are trying to identify these in a human as well, and we kind of have an hypothesis, because they have a targeted projection to the ATN, that they're involved in kind of this detonation of secondary generalization from the hippocampus, like seizure generalization to the rest. So that's really cool.
Yeah, and just the follow-up comment was that the spatial data really highlights this, because you would otherwise think that these subiculum types are resilient, but maybe there is specific subtypes that are still affected.
It's always, yeah, thanks for saying that, because resilience means not dead, but at the same time, there's so much gene expression changes happening in cells that are still there. So I think we really have to carefully remember that. Yeah.
Hey, great stuff. I couldn't help but notice in your gene load, PCA loading, that GABA B receptors are strongly downregulated.
Yes.
The field in epilepsy on GABA B receptor or upregulated, sorry, the field is kind of divided in that RNA doesn't often match the protein for these receptors. And I wonder, have you validated any of these targets with a kind of protein-based approach or an antibody-based approach to validate them functionally going forward?
So we have a couple that we are validating. Unfortunately, GABA B is not part of that. If you do have a good antibody, I would love to do that because I know some of them can be a bit tricky.
We've got some nice ones.
Yeah.
Yeah, this kind of follows on from the last the question about resilience. I mean, these, in terms of translating these changes in expression to potential interventions, let's say, should we consider whether these changes in differential gene expression are signatures of impending doom for the cells, or the fact that they're survivors, and so therefore promote or not?
So I've seen both in different cell types. So I think if we consider therapies, we have to consider cell type specific targeting, which is something that I think Ed talked about as well yesterday, or like the SCN, kind of NAV1 expression and specific inhibitory neurons for Dravet syndrome. We have to consider both, yes. That's basically my answer, unfortunately. Yeah.
I wonder if I could ask one more question. Presumably things happen in sequence. You know, there are triggering events and then sort of downstream cascades. How would you imagine approaching this in human epilepsy to actually map trajectories and selective cell loss over time?
Yeah, I would love to. So there's a bunch of trajectory analyses that are run on single-cell RNA-seq because you kind of have the introns and you can like do these trajectory analyses, which is, I think they're still being established in spatial transcriptomics, but we're working together. We have a group at UBC where you can identify one spatial spot and then see how the trajectory changes from there, which would be a graded gene expression because of cell loss in CA1, for example. So I think this would be really cool to do. And then to see if we can identify protein targets that we can label and see if we can also see this graded expression. Yeah.
Thank you again.
Thank you.